Epigenetics is a new field of science. It talks about how your environment and your experiences can change the way that your DNA works without ever changing the letters of your DNA. It has huge implications for public health. So we're going to talk about what that all means. So we spent some time doing some games related to this. So if you have never heard about epigenetics before, you are not alone. So in our previous work, most people haven't necessarily heard about it. This is for students and teachers. Now this is back in 2014, so more have heard about it since, but few can still explain it to other people. So if you are new to this area, you are not alone. So this is a graph that shows if you're looking in PubMed and scientific articles that cite epigenetics or epigenetic or epigenetically, and you can see that there's this huge influx of articles that have come predominantly recently. And if you're interested in kind of where those areas are, it's all over the place. So if you're looking at certain body systems, there's a ton related to stress, a lot related to the immune system because those are often related, but it comes up in a lot of areas as well. If you look at it related to disease systems, a lot of it is in cancer, but again, it's all over the board. So keep an eye out for it as you're looking into your various diseases or whatever topic you are interested in. So we started learning about some of these patterns related to developmental origins of health and disease. So if you have not heard of this field, look it up. It is amazing. But one of the things that I want to stress to you is the patterns that you see in science are important, and so to trust yourself. So one of the things that we use a lot is there's this region in Sweden called Norrbotten, Sweden. They kept amazing agricultural records. They kept amazing records related to deaths and births. There's a radio lab. You can learn all about it there. But what they found, there are these periods where there are either crop failures or crop abundances, and there's a strange pattern that they started to observe where the boys who had overeaten in abundant years, they tended to die a little bit earlier than the grandsons who had endured a poor harvest. And once they started controlling for socioeconomic status, so SES, it turned out to be about 32 years. They had an increased risk of diabetes death. They couldn't figure it out. Very strange. And what turns out to happen is that there are some sex-linked effects. So what they saw is that the paternal grandfather experiences predicted mortality in their grandsons. The paternal grandmother experiences predicted mortality in their granddaughters. So there seemed to be a pattern in that. And so this started to establish this importance of this slow growth period during puberty. So in girls, it's a little bit earlier. In boys, it's a little bit later. But essentially, this period is really important. The other thing that we learned related to developmental origins of health and disease is that this was this period during World War II where in the Netherlands, all of the train tracks that kind of were bringing all of these food supplies, they were cut off. So the typical food rations went from around 1,700 kilocalories a day down to around 500. So this is in the middle of winter, food isn't growing, people are starving. So I have these tulip bulbs here because people are actually digging up and eating these tulip bulbs in order to stay alive. But one of the things that they found related is that there's this increase in coronary heart disease around 50 years later. And they couldn't quite figure out why that happened. And so in this graph shows the period of the Dutch hunger winter. And you can see the increase in coronary heart disease was associated with later. So one of the things that they realized, the scientists realized as they went through, and there's a summary of this at the bottom, is that these periods, this is from conception to birth, and you can see this parallelogram going diagonally. This is that first trimester. And what they observed is this, for babies who were in the first trimester during the Dutch hunger winter, those were the babies that developed coronary heart disease 50 years later. And the reason that happens is that this is when your heart is forming. This is a critical developmental time when your heart is doing all the things that it needs to do to set up for having a functional life. So if you're not having those calories to build all of the machinery that you need to do, this is going to cause problems later. So we did this with a group of middle school boys. They also understood this process. So just to show kind of how you can describe science to the public. So I always enjoy seeing this image. But related to there are many, many organs that have critical periods for development. So the brain, from the very beginning all the way through birth, whereas other organs have very specific times. So that slow growth period that we talked about, so before puberty for girls 8 to 10 and for boys around 9 to 12. So for girls, for females, this is when the eggs are developing in utero. So for example, when I was pregnant with my daughter, it is kind of surreal to realize that actually in her ovaries, she is actually forming the eggs of my granddaughter as she is inside of me. For males, they go through puberty. Sperm development continues to happen all throughout life. But spoiler alert, there's this new science that was just released this year where they're looking at genes on the Y chromosome. It's not just sex development components, but also it's the genes on the Y chromosome are influencing the heart cell's ability to respond to fatty acids, especially in times of stress. So we're learning more and more about how our body is reacting to stress, how we can, and what that means for health systems. So there are other patterns that continue to emerge. So for example, the Great Leap Forward. So this was in China right around 1958 to 1960. And so this is where they were trying to move from agriculture to an industrialized society. And so they were trying to get folks to leave their farms and really support industrial revolution. During this period, about 20 million people died of starvation. So this is a period that I imagine that you'll start to see more and more of as we continue on. So, and the things that they start to see is of all of the outcomes, associations between the early pre-birth famine and adult body size. You see issues related to diabetes and schizophrenia. Those are the most consistent patterns. But there's so many other things that they're starting to observe. So keep an eye on this period. And also, skinny does not mean healthy. So we talked about the Dutch hunger winter. So Audrey Hepburn went through that period. So at 16 years old, she was 5'6" and weighed about 88 pounds. I know that she is celebrated for Breakfast at Tiffany's and other wonderful movies. However, she really suffered health-wise all through her life. Famine and malnutrition have huge problems that can produce health effects. So epigenetics is a way to change the volume of gene expression. So I like to think of it as like a car radio, for example. Your car radio is always there, regardless of if you're driving, if it's turned on or off. Now, you can turn up that volume, you can crank that music and blast it really loud, or you can make it very, very soft. This is the same thing with gene expression. Gene expression is simply how your DNA controls the protein output that's coming from your body. So we're going to go a little bit more into that. So when we think about, say, a genetic mutation, that is actually changing a letter, say, going from a C to an A. So something is actually changing related to the genetic code. Epigenetics is a little bit different. It's not changing any of the letters. However, what it's doing is it's making a little chemical tag on it. So it changes the way that the DNA is read, and that is a huge difference. Okay. So if you think back to high school biology, you may remember that it goes typically, theoretically, from DNA to RNA to protein. So this is called from transcription, so DNA to RNA, and then translation from RNA to protein. So this is the scientific, a quick diagram that kind of shows you what that looks like. But ultimately, you'll get some protein that comes out of that, and that protein is kind of everything that makes your body function. The DNA may be the instruction manual, but the proteins are how you work. So what's interesting is that the proteins are decided based on the DNA sequence. So for example, based on the order of the DNA, it will spit out or it will translate a different amino acid that will make up what that protein is made out of. So if you're changing the letter of if you're changing the letter of your DNA from a genetic mutation, this is where a different amino acid is coded and you get a different protein as a result. The shape of the protein influences how it works. So spoiler alert, that makes a big, big, big difference. But with epigenetics, it's different. So again, you have this chemical tag. So it's not the same exact amino acid is coded. So it's the same protein that gets coded. But it's all about the access and how the transcriptional machinery is able to read that. So when we're looking at epigenetics, so this is a view of your DNA, how it's folded, and you can see that you have that double helix. And then as you start to get really tightly wound, it wraps around these histones. Those are the gray things that you see right there. And it goes really, 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 really tightly into that X that you see off to the bottom left. And that is your chromosome. So in order to be able to read all of that DNA, you need to be able to unwind and wind it back up so you can fit it into the cell and fit a bunch of information in there. So there's your chromosome, there's your histones. But what's pretty cool is that when you're looking at epigenetics, those chemical tags that we talked about, those are just simply methyl groups that get added to the DNA. And this can help change the way that the DNA is read. So for example, if you're trying to reduce gene expression or silence it, turn it off, you can methylate it. So this makes the transcriptional machinery and the translational machinery harder to read that DNA. So then you're not able to get that same code. Likewise, your DNA is around really, really, really tightly around histones. So if you wrap something so tightly where you can't even read what's on there, it makes it very hard to read that genetic sequence. So putting it very, very tightly makes it harder to read. Alternatively, you can increase gene expression by either removing the methyl groups, or you can acetylate the histones, and that just essentially, it's based on the positive-negative charge. It releases the DNA from the histones and makes it easier to read. This is how I remember it. I think of it as a skunk, so methyl groups can be smelly. And so I think of methyl as in skunk, and the skunk is something that makes you want to go away. So this is, methylation typically turns things off. Just a mnemonic. This is not always the case, but again, as we're starting to make patterns, this is something that works for me. So we often get the question, is reducing gene expression good or bad? And it really, really depends on the context. So for example, if you are climbing Mount Everest and you need to acclimatize to the elevation so your body is able to transport oxygen, this is a really good thing. You need to do that, and that helps you for your body to better respond to exposures such as low oxygen and cold stress. However, it can depend. So if you're talking about the cell cycle, normal cell growth, we need that. So this is how we form skin cells, this is how we form scabs. So that epigenetic process is wonderful, we need it. But if you're looking at, say, a tumor suppressor gene involved in cancer, by stopping that tumor suppressor gene, by methylating it, for example, you are essentially turning it on and having it go longer. And so this is where you start to get a bit abnormal cell growth. So that might be considered bad. The other way that you can think about it is in the context of behavior. So this is all about bees for this next little bit, but one of the crazy things is that we've learned so much about epigenetics because of these bees. So that when you think about bees, know that in a hive, around 90% of bees in a hive are worker bees. And those are typically either the nurses who take care of the baby bees, they build the nest, and also the foragers. They go out, they collect the pollen, the nectar, everything that the bees need for their colony. And they work together. So as you're looking at these nurses and foragers, they're genetically identical. The only difference between them is actually the nurses have different segments that are turned on versus off, and this is all due to epigenetics. So as their role in the hive, this is something that I find completely amazing every time I hear about it. So just after birth, most of the bees become nurses. They need bees to take care of the young. However, as they get older, more and more become foragers. But what turns out to be extremely fascinating is that if you reduce the amount of nurses in a hive, then those foragers will actually revert to become nurses in the hives. And again, the only difference is this epigenetic sequence back and forth. So behavior makes a huge difference, and epigenetics allows us to translate what's happening in the environment to behavior. Diet is also having a big impact. So for example, the queen bees that we hear so much about, they are also generally genetically similar. The difference is that many of these larvae who are bathed in this royal jelly. They don't technically eat it, but they are bathed in this royal jelly, which is produced by the nurses. They are the ones who develop into queen bees later. And what's fascinating is that as long as the queen bee is alive and functional, she's producing pheromone, and so nurses know not to produce more of this royal jelly. However, once that pheromone starts going away, that is when the nurses know to produce more royal jelly so a new queen can be developed. You'll also hear about it related to the agouti gene. So this is one of the best understood epigenetic related examples. So this is through Dana Dolinoy's lab, and they looked at, so these two mice, well, although these are cartoon versions, these two mice are genetically identical. The only difference between them is that the brown mice, which is considered the typical mouse, has their agouti gene that is turned off through methylation. The yellow mouse, which is typically a little bit more obese, has that agouti gene turned on. If you look at the pups of these animals, and actually before we do, here's the variation. So between the agouti mouse all the way to the left and then the brown mouse all the way to the right. So again, genetically similar, just different levels of agouti epigenetic markings. So if you look at the pups of these mice, the brown mice typically have pups who are also brown, considered considerably lower in body size, whereas the agouti mouse has the yellow pups associated with that agouti phenotype. If you look at those pups, they are genetically identical as well. Same thing though. So the brown mice have the agouti gene turned off. The yellow mice have the agouti gene turned on. Feeding the agouti mouse many fruits and vegetables, specifically ones that have a lot of methyl donors, you can actually turn off that agouti gene, and their pups then become these brown mice associated with this off agouti gene as well. So there seems to be this pattern between diet and epigenetics that is really, really fascinating. One of the new areas of science that we're starting to see is this social interactions and social epigenetics. It's a fascinating field. We are learning so much more about this. I highly recommend it. And so we're learning that not only some of these historical threats, like physical threats of violence, but also some of these social threats. So all of the things that can really, really stress us out, they can influence our bodies. So this may happen through the immune system. So this is what they're starting to look at right now. So this could be either wound repair and response to bacteria, or this could be inflammation-related diseases. So watch for this. It is a fascinating, fascinating area. Related to epigenetics in the social context, one of the earliest studies looked at the relationship between methylation of genes and maternal behavior. So there's a fascinating story about how they discovered it. Read it. They were not expecting to see the results that they did. They were fascinated to see it. But what they're saying is that maternal behavior can alter the stress response of the offspring. And the way that they studied this is that they're typically attentive mothers, rat pup mothers, and inattentive mothers. And they looked at the genes of the brains of their pups, and they saw that there's differences in the genes that control stress. So for example, when the genes in this region were highly methylated, they saw increased stress in the pups, whereas if they were rarely methylated, they saw lower stress. In their second experiment, what they did is same thing, but they just switched the mothers and the pups. So the attentive mother raised the attentive mother raised the pups that were born from the inattentive mother and vice versa. And what they saw is that the maternal behavior had a huge impact on that. So they see the same thing as they saw in experiment 1, but just by changing the environment. Finally, in their third experiment, all they did was they injected a drug that removes methyl groups into the brain. And then they saw this disappearance of some of these epigenetic changes in these rat pups and a lower stress response. So this was one of the very, very first ones. They've been looking at this since, but know that there's some relationships, some strong relationships between stress, environment, exposures, And how that looks is going to differ a lot depending on what exactly you're looking at. So in public health, we see so many patterns. So think about these patterns. So if epigenetics is a way for thinking about health, and we know that stress and diet are the strongest influencers so far, think about how traumatic experiences may be manifested in some of these epigenetic markings. So it could be now through various wars, it could be through the Holocaust. There's some evidence of civil war. This could be related to massacres and famines. This is ethnic displacements and migrations. This could be social, like being raised in abusive environments, bullying, and there's some research on suicide in that area. And then I teach environmental systems and human health, specifically environmental health concepts and environmental health. And the effects of pollution and toxins are huge for us. Same thing with wildfire and flood displacements that we're seeing all over the country due to climate change. And even right now, related to COVID-19, we're seeing food shortages and food banks, and I imagine there are going to be additional stressors as our food systems get stressed. So as we're thinking about all of these components, how might these experiences influence the epigenetics of our populations? There's some new research that's looking at social stress and the impacts on inflammation. So I am a huge fan of inflammation. It is something that I've studied previously. But as we're thinking about some of these things, so social isolation is having a big effect on how our immune cells are functioning. So as we're looking at this, be mindful of that. Also, another thing that's starting to happen is that we're realizing that there are massive racial and ethnic disparities in the adverse social exposures that these populations are experiencing. So this is from Alexandra Shields, who is a PhD out of Harvard. She came to OHSU this summer, and she gave a beautiful talk on some of these exposures. The thing that I would like you to take away is that her research is amazing because she's looking at the epigenetics and how that relates to health disparities. So if you need a quick review on the topic, please see the one in orange. It's lovely. But what she's seeing is that some of these DNA signatures in the methylation, they are still powerful and stable after years of abuse that happened way earlier. So on this right-hand side, she's looking at how childhood abuse, either the frequency abuse, which is the first one up at the top, or the severity of abuse, which is the one at the bottom, can influence the methylation of NR3C1, which is at the bottom. This is a stress response gene, so it tells how it works at the bottom. But as we're thinking about this, really be mindful of how do these social exposures that are horrible, how are they influencing some of our epigenetics and how our body systems work. And I want to also orient you to her work because she has some of these beautiful studies related to how racism can influence telomere length and how you can use emotional support to actually improve some outcomes. So she works in a transdisciplinary team that really looks at stress and spirituality, so in a non-denominational way. And she sees some amazing results. Black and Hispanic Latino communities are highly spiritual, according to her research. And so by working with these communities in areas where they are getting the support that they need, this might be a way that we may be able to reduce some of these epigenetic effects that we're seeing. So as we go through, I am expecting to see more and more epigenetics related to all of these social exposures, related to racism, related to social injustice. We're going to likely see more of that. So keep an eye out for it. The other place where we're starting to see some epigenetics is related to copying errors in cancer. So there was a study that came out in 2017 that was looking at cancer incidence, maybe related to cell division and in how the body is copying the DNA, and maybe there are some errors in there. So there's a video that you can also watch that tells more about this. But there's some interesting components here. So thinking about how epigenetics may be involved. And it may be one of the reasons that we see so much epigenetics in cancer specifically. So as you think about this process, here are a couple things that I want you to keep in mind. One is the environment affects our genes, and you might have a lot more control than you previously thought. There is so much that we don't know yet. It is a cutting edge area of science, so keep it in the back burner of how things are working. But it's likely to have a big role in a lot of areas. So this could be learning and memory, sleep, disease risk, and developmental origins of health and disease. But the one thing that, as you're talking with the public about how epigenetics work, they really want to stress that really be careful to not focus on the specific mechanisms, because those are likely to become outdated as the science progresses. For example, it's not necessarily your job to get bogged down in the details of how this works, but to teach them the concept that the environment and our exposures to various things in our environment can change the way that our DNA work. So for example, we already know that there are new mechanisms for producing epigenetic effects. So there are a few down below. And non-coding RNAs are just a fascinating example. So we have our exons and introns, and as it goes from DNA to RNA, it removes the introns. So what happens with all of those introns, and how do they actually help to regulate what is happening in the body? So completely fascinating. So again, remember the patterns and simplify. So some things to keep in mind is that every tissue in the body has its own epigenome, which can be both overwhelming and allow you to help make some predictions. So if you were to simplify, based on what that organ does, what would you predict those outcomes to be? But ultimately, the reason that I love epigenetics so much is because it really gives a science for why a person's choices matter. And it really describes how science is constantly advancing. So there are so many things that we didn't know previously that we now know is actually having a huge impact. And because epigenetic marks are so stable, it allows us to measure them. So we were able to do some beautiful studies between epigenetics, social stress, and environmental exposures, and so many other things. So as we're thinking about this, keep these in mind. So one thing to be concerned about is be very wary of, be critical of how you're seeing some of this epigenetic information being presented. So are there new products that are claiming to do certain things? Are they overreaching interpretations? So these are things that we'll have to be critical of as we move forward. When we were doing the epigenetics project, we have a teacher advisory board who helped us to kind of think about what to consider when teaching the topic to the public. They have three primary concerns. One is social determinants of health. So as you're introducing a topic, be mindful that individuals from disadvantaged backgrounds, especially students who may be younger, they may not have the ability to change their diet or stress or where they live due to socioeconomics or many, many other super reasonable factors. So be mindful of that. There's also some ethical and social justice considerations. So these topics can be really sensitive for some minority groups. So for example, there is a study looking at a Holocaust survivor offspring. And also that teachers are largely unprepared to teach the topic. So many of them at the time were unfamiliar with it, and they need materials to help them do that. So what they really, really encourage is to focus on the cutting edge aspect of epigenetics. And rather than use personal identities, instead use avatars or characters to really describe critical periods. And finally, if there are briefing sheets or packets that can help, that is also wonderful. So as you're talking about some of these things, if you can have your students or the people with whom you're working be able to recognize the term, that is gold. And if they can understand that epigenetics is a way to modify health outcomes that can go in both directions, that is also wonderful. And if they are able to recognize the term and know that it can change health outcomes, then this is some of that process of science that will make learning about some of these new areas way more fascinating. So one of my favorite articles is at the bottom. So this is Social Epigenetics, and they were talking about how two people can be in the exact same room, in the exact same environment. But if one is extremely stressed out because of the other person, those two individuals are going to have very, very different experiences as a result of that environment. So this is a lot of what epigenetics is. If you need any resources for either genetics or for epigenetics, these are some that I love a lot. There are also, I have run a program called Let's Get Healthy that has online resources to teach it. So there are interactive digital media for it. There are all sorts of lessons and activities, and there's many, many different manipulatives and other. You can find those here. So you can have DNA and you can put chemical tags on it, which makes it harder to read. Or you can wrap it around a fuzzy stick, also previously known as pipe cleaners. And so that can show the wrapping around histones. And I really like the cookbook analogy. So, for example, you have your grandmother's cookbook, and the way that you alter it is your epigenetics. So you're making additional marks to change the way that it might be produced, even though you still have all of the original text, which is arguably your DNA. Alexandra Shields uses a sheet analogy music where you have the original score and then all of the annotations that are made on top of that. So as you're thinking about these things, there are many beautiful ways to really kind of think about epigenetics and teach it to the public. But ultimately, as you're doing this, think about it as a knob of a radio where we can turn it up or down, and that is how our body can work.